Why RNA World does not work for abiogenesis. 1. The RNA World Hypothesis proposes that abiotic RNA molecules created self-replicators as a precursor to DNA and protein-based life. While this hypothesis is widely believed, there are several scientific arguments against it. 1. Chemical complexity of RNA. The formation of RNA molecules requires a series of complex chemical reactions that are extremely unlikely to have occurred spontaneously on the early Earth. The ribonucleotides that make up RNA are themselves complex molecules, and their realistic formation under realistic early Earth prebiotic conditions is empirically impossible in sufficient and controlled quantities needed for RNA origin of life. 2. Instability of RNA. RNA is inherently unstable and prone to degradation, which provides strong evidence against its ability to function as a stable genetic material in early life forms. This instability would have made it impossible for RNA to accumulate in significant quantities and persist over time in the harsh conditions of early Earth. 3. Catalytic limitations. While RNA can act as a limited catalyst, ribozymes, its catalytic capabilities are very limited compared to proteins. The range of chemical reactions that RNA can catalyze is much narrower than that of proteins, which would severely limit any complexity and diversity of early RNA-based life. This is strong evidence against the RNA world for origin of life. 4. Problem of chirality. RNA is made up of chiral molecules, meaning they exist in two forms that are mirror images of each other. Prebiotic synthesis of RNA would have produced equal amounts of both forms, but life uses only one. There is no naturalistic mechanism to select just one form for the RNA world, so this is a showstopper for RNA world origin of life. 5. Transition to DNA protein world. The transition from an RNA world to a DNA protein world involves complex biochemical changes. DNA is a more stable repository of genetic information, and proteins are more efficient catalysts. However, there is no realistic naturalistic mechanism for the formation of these molecules and their integration into cellular mechanisms in a realistic early Earth environment. This is a showstopper for RNA world origin of life. 6. Unrealistic experiments. While there have been some laboratory successes in demonstrating some low-level self-replication and catalytic activity in RNA, these experiments are conducted under highly controlled, idealized conditions that do not accurately reflect the environment of the early Earth. Realistic environments do not provide the required results. 7. Lab experiments. These confirm the need for intelligent design of the specific conditions and changes in conditions in correct and timed sequences for the desired molecules. This points to the need for intelligent design for the origin of life. 8. RNA's complexity and origin. The synthesis of RNA requires relatively complex prebiotic organic chemistry. The origin of such complexity in a prebiotic world, by purely naturalistic means, is a critical barrier, standing as evidence against the notion that RNA was a prebiotic self-replicating molecule that led to life. Additional details. 9. Prebiotic synthesis. RNA is a complex molecule with 4 different bases and a sugar-phosphate backbone. The conditions on early Earth would not have been conducive to the formation of RNA in sufficient quantities for the reactions needed for RNA world origin of life. 10. Catalytic activity. For the RNA world to work, RNA molecules would need to have had the ability to catalyze essential chemical reactions. While some ribozymes, catalytic RNA molecules, have been discovered, their range of activities is limited, and they often require specific conditions to function. Their catalytic capabilities are not sufficient for RNA world origin of life. 11. Instability. Hydrolysis. RNA is susceptible to breakdown by water. This short lifespan would have made it impossible for RNA to accumulate and evolve in the early Earth environment. 12. Fidelity. Replication of RNA is error-prone, leading to mutations that would destroy its function. So, RNA would not have maintained the necessary level of accuracy for long enough to evolve into more complex systems for RNA world origin of life. 13. Genetic code. The origin of the genetic code, which dictates how RNA sequences are translated into proteins, is not a capability of the RNA world, so this stands as evidence against RNA world origin of life. Conclusion. 1. RNA world does not have the ability to create life by naturalistic means. 2. RNA world does not work for naturalistic abiogenesis. 3. RNA world is the best of the models that is available. 4. That means that none of the origin of life models work for naturalistic abiogenesis. Thanks for watching. Please like, share, and subscribe.